The bioprocessing industry is continually challenged to increase productivity while keeping the cost of goods down when developing the next generation of therapeutics. Now, one key aspect of the downstream purification process is the capture step, which relies upon protein A chromatography resins. I'm Miles Skoda. This is the Life Science Connect webinar series, and today we're addressing the challenges of complex biologic purification, new advances in protein A affinity resins. Now, joining me for this presentation are two industry experts from Purolite, Andy Masters, Head of Business Development and Strategy, and Mark Hicks, Research Development and Engineering Group Leader for Ecolab Bioprocessing, who together will discuss modern challenges in protein A purification, how strategic partnerships are benefiting technology innovation, and Ecolab's affinity toolbox of chromatography resins. Now, we'll be holding a Q&A session following this presentation. Please submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we don't get to, we'll follow up on after the event. And then just a bit of housekeeping: if you run into any issues today, please try refreshing your browser first. If your problem persists, submit a question through that Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day, often less. You'll get an email with a link to it, or you can just use the same link that you used today. So with all that said, I'm going to turn it over to Andy to get started.
It's a real pleasure to be involved in presenting one of our first webinars as part of the newly formed Bioprocessing and Technology divisions of Ecolab. For those of you who are not aware, Ecolab acquired Purolite in November 2021, and now as a fully integrated division, it's excellent to be able to highlight at the start of this webinar some of the commitments that Ecolab have made already and plan to make with regard to our long-term success and the desire to expand Ecolab's presence across the bioprocess industry. I think when we look at the investment that Ecolab has made following the acquisition of Purolite, we can look at three main areas. And the first is a significant and ongoing investment which has been made to expand the global capacity and resin supply. With that investment, we're really currently expanding our UK facility, which is based in Wales, where we make the agarose-based beads and the resins. And we're also gearing up for an additional facility in the US. This will have a dual impact, not only to increase our standard supply, but also to support contingency planning for our customers. In line with that, we've taken the opportunity to actually align with Ecolab business systems, so we'll be able to give our customers more visibility into our supply and our inventory. Second to that was the product innovation, and I think it's one area where Ecolab have focused from the very get-go after acquiring Purolite, and that's been highlighted really by the recent launch of DurA Cycle A50, which is a new protein A resin launched at Bio US early this year, and it marked the third product that we've launched in the last 18 months. So this really shows the direct and significant impact of Ecolab's investment in Purolite R&D. This also expands our current toolbox of purification resins and really allows us to facilitate the purification and manufacture of a wide range of molecules, Fabs, FC fusion proteins, bispecifics, and other complex antibodies that are part of our customers' product pipeline. Some examples of this you will hear more about later from Mark during the technical aspects of this talk. Finally, Ecolab has recently finished work on the bioprocess applications laboratory in the US at our King of Prussia site called BPAL, where we've installed a state-of-the-art lab, high throughput process development robotic systems, bench and large-scale chromatography columns and systems. And the site itself is staffed by experienced scientists who were actually customers themselves, who understand the customers' challenges, who understand how to carry out the process optimization. And together with the field application specialists, Ecolab can now offer comprehensive support and training of our bioprocess customers. So after setting the scene with regard to some of the investments that Ecolab have made, I really want to talk a little bit about some of the challenges that we're seeing in the processing industry for monoclonal antibodies. I think it's worth covering these because this is the sort of information that we see from conferences, but also get from customer feedback. And again, I want to look at this in three pillars. The first one really is the adoption, or at least the evaluation of new processing models. And I think it's fair to say that within the bioprocess industry, They're renowned for being relatively slow to change established ways of working, but we are seeing an ever-increasing focus towards process intensification. In my experience in existing facilities, possibly due to easier implementation and facility fit, this usually starts with the intensification of an individual upstream or downstream unit operation. For example, you see people switching from batch expression to perfusion expression, depending upon the application, or it may involve the implementation of multi-column chromatography, rapid cycling, or a continuous affinity capture step. This tends to see that we're seeing more connected processes with the end goal really of a continuous state or fully automated manufacture of antibody therapeutics. I think this trend places additional demands on vendors, not just only from a system and column perspective, but also from vendors who are manufacturing resins. And I think there's a desire to have resins with an increasing level of mass transfer, which allows high capacities at short residence times, which can drive this productivity. But they also want to see improvements in the stability of resins to maximize the lifetime and then combine and drive down the cost of goods. In parallel to this, there is clearly a shift in the molecular pipeline of many of our customers, with an increased focus on development of ADCs, antibody drug conjugates, and in particular, these multi-specific formats. And I think, bispecifics are a good example where we've seen growth of double digits in the last few years, and they are due to increase even further going forward. By their nature, they've increased in complexity over standard mAbs. So what we're tending to see is developers pushing the capture step even harder than before. Again, I think it's probably fair to say that most of our customers are utilizing a toolbox approach, which contains a number of affinity resins with novel selectivities, where the capture step is not only screened for capture of the target molecule, but in its ability to remove closely related byproducts, which then can simplify the subsequent polishing steps and build overall robustness into their process. I think a really good example of that is a recent 2024 publication in the Journal of Biological Methods, which discussed the unique properties of a resin called Presto CH170, which is an Ecolab resin, which was developed to bind to the human CH1 domain of IgG. However, due to the unique properties which they found within their study, it allowed them to not only capture a bispecific, but to separate the homodimer from the heterodimer species in the purification of these asymmetric bispecifics. As well as these selectivities, I think newer ligands and resins are evaluated not only just for this selectivity, but also what ancillary benefits can they get from it. For example, can we push the resin to increase the recovery of mAb monomer? Can we improve the removal of host cell proteins? Or can we leverage different conditions to actually perhaps look at protein A for increasing the log reduction in a viral clearance study? Some of these challenges will be discussed in a lot more detail and described by Mark later in the talk. And finally, the supply chain. I think most of us, with COVID, it just seemed like a bit like a distant memory. However, the impact on the bioprocess industry was huge. And most companies have or plan to reassess their supply chain. I think a lot of companies talk now about strategies for a wider dual sourcing of critical raw materials, not just for instance, for a filter for buffers, but also for products which have a closer contact to the product themselves and have an impact on the critical quality attributes. This has been supported by industry bodies such as the Bioforum Organization, or BPOG, and there are many publications now highlighting some of the regulatory aspects to support this sort of strategy. I think we all agree, that purification performance of a resin is key for its selection as part of a process, but it is important, and we are aware, that we're part of a risk-averse industry. So understandably, there's a focus on quality and long-term supply of critical raw materials throughout the lifetime of a drug. And I think this is something that Ecolab is fully aware of, and hopefully I highlighted in the commitment through the recent investments. I already mentioned that at the beginning of the talk. But Ecolab has always wanted to commit to drive innovation and build on the relationship with Repligen as a partner. Ecolab and Repligen have been working together since 2017, and to date have commercialized approximately 8 affinity resins, but always trying to improve the performance and the range of selectivities. I think critical to that success has been the development of jetting technology for the manufacture of the agarose beads. This method not only produces highly uniform bead intermediates, which do excel in their mass transfer properties, and we've had feedback from customers who evaluated them, but they're also manufactured with a minimal use of organic solvents and sieving compared to emulsification methods. This results in a shortened manufacture and subsequently a shortened time for lead times. But key to this really is the ability to remove the need for the use of organic solvents, or reduce that need and reduce overall waste. Ecolab itself is also recognized as the leader in sustainability, and the use of jetting technology aligns with our sustainability goals, but also supports those sustainability goals of our customers. We tend to be focusing now on how raw materials, reactants, and buffers contribute to their process mass index, and in particular water consumption, which is a key concern for the bioprocessing industry. Repligen are also a global leader in ligand design, but just as importantly, they have the ability to scale up and supply high quality ligands at the quantities needed for bioprocessing products. And that's important to Ecolab, who established an excellent partnership to deliver innovative affinity resins to the market. And that's not just established protein A products such as Jet A50. In the last 2 years, we've brought a protein A resin called Jet A50 high pH and a CH1 binder resin, which was specifically designed to address some of the purification challenges for these new antibody formats that our customers are developing as therapeutics. On top of this, Repligen have a well-established record of supplying pre-packed columns, and together with the combined experience of Ecolab scientists and our field application services, we can now support our customers from process development to full-scale manufacture. So with that short introduction, I would now like to pass over to Mark Hicks, who will give a more in-depth insight into the latest resins launched by Ecolab as an outcome of this partnership. Over to you, Mark.
Thanks, Andy. It's a real pleasure to talk a little bit more of the technical side of our latest innovations on protein A resin development for chromatography. The first resin that I'm going to walk through some data on is our latest protein A resin, which is called DurA Cycle A50. DurA Cycle A50 was developed to meet a lot of the current challenges that Andy highlighted earlier in the talk, with the improvement in upstream, the change in molecules. You've got higher titers, you've got higher complexity, brings along with it its own challenges. One of those challenges is you need harsher cleaning conditions, or they're required sometimes in between. Standard protein A was designed more for 0.1 molar sodium hydroxide stability, and then there's a limitation there as well. With DurA Cycle A50, it's been designed to be a high capacity resin that actually is more stable to a lot of these conditions. So as I mentioned, sodium hydroxide is one of the key areas that we need to look in terms of stability. It's the most common cleaning solution. There is more of a drive in the industry to go up to 0.5 molar sodium hydroxide as standard. So that's been developed with this new resin. But when we talk about stability, there's also the other aspect that needs to be considered, which is the stability to the feed itself. Each feed is different. Each feed is going to be containing multiple enzymes, multiple different proteins. And these can have challenges with protein A. They can enzymatically clip the ligand, which affects the overall stability of the resin, and they can be very different feed to feed. So having a resistance to those is really important. Mild pH solution is something that we've previously talked about with the release of Presto Jet A50 high pH, and I'm going to talk a little bit more about that later in the presentation. But that's a unique property of one of our innovations, and we've incorporated that into this resin. And finally, every protein A resin has to be a high capacity resin. Your productivity and how much antibody you can deliver is all linked, how much you can load on the Protein A resin. So keeping cost of goods low is really, really important, and you need high capacity for that. So as I mentioned on the previous slide is the importance of high capacity. You can see with the graph on the right, which actually looks at polyclonal IgG, that DurA Cycle A50 overlaps the previous generation resin that we developed, PrestoJet A50, which is a very high capacity resin. And this has been shown to be the case with other MABs that we have access to. We've done IgG1, IgG4, and they're all showing high capacity and good overlap with Jet A50, which is a leader in the market when it comes to capacity. So it's really, really important that we keep that capacity or better it. It's on the 50 micron bead, the same 50 micron bead that Jet A50 and PrestoJet A50 high pH are on, which gives you that uniqueness in terms of jetted resin and greater mass transfer, the higher flows, gives you that flexibility in terms of process intensification by monitoring your bed height and your diameter to get the best out of your resin. And the other aspect is it's all developed on the same bead. So flow expectations are the same across the range. And talking about those flow expectations, there's two graphs here which show pressure flow at a 30 centimeter ID and a 60 centimeter ID. There's also data available on much higher scale. We've also got customers using Jet A50 up to 1.6 meter columns at 20 centimeter bed height. It can be expected to get up to 200 centimeters an hour at scale, at production scale, at a 20 centimeter bed height with Jet A50. But one thing to highlight on this slide is legacy type columns which pack by pressure or manual compression really do result in a higher degree of compression because of the nature of the compression being layer on layer and the bottom being more compressed than the top. Axial compression was really designed for these smaller, softer beads. And so by having that more continuous pack, you do end up having a looser pack, for want of a better term, which allows you to get a little bit more flow out. So we would recommend those who want to use the 50 micron bead after to get the most out of the flow to use axial compression. That doesn't mean you can't use pressure pack, but you'll get better flows with axial compression. So here we're showing some stability data where we've exposed the column to 0.5 molar sodium hydroxide. This is non-protein or non-feed loading. We've run it as cycles using a buffer-based blank loading phase and then used the same CIP conditions that we would do during cycling. And what you can see in pure sodium hydroxide stability, it's matching the market leader in terms of alkaline stability. But I think the key in terms of development and innovation, if we look at the light blue line, which is PrestoJet A50, which was not designed as a 0.5 molar sodium hydroxide resin, the new engineering on the ligand has significantly increased the stability. So we're addressing the need for a more stable Protein A resin to be used with these newer molecules, newer challenges, newer processes. And still on the topic of stability, we mentioned earlier talking about protease resistance and the impact that the feed has with proteases and other proteins which can cause damage to the Protein A ligand, which and ultimately affect the product lifetime. Which you've got in the graphs here is you've got three molecules that we've tested. All three of these molecules are high protease challenging feed streams. Even the monoclonal antibody, it was from a different cell line to what was normally produced on this antibody, and we were looking at the impact of that. And you can see with Jet A50 that increase in protease stability did actually affect the ligand stability. Jet A50 high pH, which is the next generation resin, has an improvement again on that stability, but DurA Cycle A50 has been specifically designed to be resistant to these enzymatic cleavage points, and you can see that it retains stability after 60 hours exposed at 37 degrees to these antibodies. So it's very extreme conditions, and you can see that the stability is really there. So having the stability to the feed, but also the sodium hydroxide, gives that increased stability, increased expected lifetime, increased ability to clean the resins with harsh conditions, which is often needed or implemented as a standard process. And then we're going to focus on the uniqueness. As we mentioned before, PrestoJet at A50 high pH has the ability to do milder pH elution. And I'll go on later in the talk and show the positive impacts that has on the overall quality attributes of the molecule being used at milder pH. This unique property is partially engineered into DurA Cycle A50 as well. And so you can see with the graph that's being shown, you can get quantitative elution up to pH 4.2. Whereas if you look at a traditional protein like jet at A50, by the time we're at pH 4, the recovery is significantly dropping off. It's normally used 3 to 3.5. So that was the introduction of DurA Cycle A50, our latest generation resin, which was designed for high stability, high capacity, but also has this unique behavior of mild pH. I'm going to switch focus now to really focus on that mild pH elution and what are the benefits. And I'm going to be looking at case studies using PrestoJet at A50 high pH. As a recap, this resin is a protein A resin. It's designed as an FC binder, and it has the ability to elute up to pH 5 without the need for any additives. And there are multiple benefits that this can have. What we'll show in the later case studies is the effect this has on the overall process impurities profile. This can not only be really beneficial to molecules that previously couldn't use protein A because of acid sensitivity, for example, but also standard processes where it can reduce the burden on the subsequent chromatography steps. Why is that important? All the impurities that come through can have a detrimental impact, not only to the resin, which is what we're focusing on, but more specifically to the actual product quality, which can make it unsafe, can put patients at risk, can put manufacturers at risk that the product needs to be failed and can't longer process. It can reduce process efficiencies. If you can reduce the burden on subsequent steps, you can start to look to, well, can I optimize that process? Do I need to run it as long? Do I need the intermediate steps between them? You can start to think about optimizing those. And the whole process intensification strategy becomes a holistic one if you can start to improve the performance of each step. It helps with the regulatory and compliance. As Andy mentioned, things have changed since COVID, and there's BPOG requirements and others around there that really focus on proving critical quality attributes rather than identical validation process between processes. So when you focus on critical quality attributes, if there's any way you could improve them, reduce them with less steps, reduce them with an optimized process by mild pH elution, there's of a huge benefit. And then, as we mentioned earlier, you need to reduce these impurities. They can have dramatic effects on patient safety and efficacy of the drug. So anything we can do to improve and optimize that process by our innovation with resins is something that we're always looking at and always want to challenge, as well as contribute and collaborate with our customers. So the impurities assessment has been done over 3 case studies, which I'm going to go through later on. We're looking at 4 different attributes. We're looking at high molecular weight content or aggregation, really looking at stability of low pH in that case. We're looking at host cell protein carryover, residual DNA carryover, and viral clearance. And in this study, we'll compare jet at A50 high pH. We've got our traditional protein A resin, which is PrestoJet at A50 in these cases, and then we've compared them against competitor A, competitor B. Competitor A is a widely adopted agarose protein A resin. Competitor B is a high performance latest generation agarose protein A resin. And you can see with case study 1, we've looked at 3 molecules. We've looked at a monoclonal antibody for IgG1 subclass and an FC fusion protein. These are fairly stable molecules, but they have different challenges in terms of host cell protein and DNA clearance. And we've looked at a biospecific antibody, which is acid sensitive and has stability issues. All the standard protein A resins are run at 3.5. With the MAB and the FC fusion, we were able to optimize to 4.5, and with the biospecific to 4.8. Case study 2 and case study 3 look at viral clearance. Case study 1 does mock viral clearance and uses an IgG1. Again, traditional protein A is at pH 3.5. But with high pH, we've started at 3.5 and then gone up in 0.5 pH increments, as long as we could quantitatively elute the product. And then case study 3 is a live virus study that a contract manufacturer has done on our behalf. This uses an IgG4. Again, traditional protein A was using pH 3.5 and high pH we used 3.5, and we were able to optimize at pH 4.5 for the elution. So as you can see from case study 1, the graph on the right, with aggregation and sort of stability of the molecule, there are small improvements that can be seen with the Fc fusion protein and the MAB by doing a milder pH, but these are relatively stable molecules. So in these cases, stability, high molecular weight issues wasn't necessarily a big target. But with the acid labile molecule, you can look at the bispecific antibody. This process would currently be uneconomical with over 40% high molecular weights. It wouldn't be a process that would progress. You'd have to do significant engineering. But you can see just by being able to elute at a milder pH, you're stopping that point at which a large degree of aggregation begins with low pH. You can reduce it by almost 20%. That makes this process now economical. With some upstream optimizations, you could make this process going forward. Previously, this would be uneconomical and wouldn't go forward. So you can see the benefit in terms of stability for molecules that are acid sensitive, but also other molecules. And then we look at host cell proteins, and what you can see by all 3 molecules, that there is a significant increase in the log reduction of host cell proteins by being able to elute at a milder pH. So you can see that disassociation between the antibody and the host cell proteins is greater by being able to elute the target molecule at a milder pH. And we see exactly the same behavior with residual DNA. There is a higher degree of disassociation between DNA and the target molecule. And the reason and the theory behind this is by creating a more favorable condition for the antibody to elute by the engineer of the ligand, it's putting it in a condition that is actually unfavorable for the host cell proteins and DNA that have associated themselves with the antibody to elute. So you get greater disassociation and greater separation. And we're going to look at that also with viral clearance, which should show the same behavior. So case study 2 and case study 3, on the previous slide mentioned, they're labeled 1 and 2 here. We're looking at viral clearance. The first study that we do is MOC-V, it's using retrovirus-like particles and an IgG1 MAB. And the second one is live virus, where we used enveloped and non-enveloped viruses via a contract manufacturer using an IgG4. So on this slide, we're just highlighting the viral clearance approach in terms of downstream purification. The overall process is holistic. It's an overall reduction, so it's a contributing factor from every step. It's part of that downstream, and you need a minimum of 40 log reduction in viral clearance for any drug approval. Protein A is normally 2 to 3 logs contributing to the viral clearance. So any increase that we can do in terms of log reduction values for an individual step reduces the burden on the overall process. And the question then is can you improve it enough that you actually reduce the burden so much that you can reduce operations? You don't need as many. That can have a huge cost saving and process optimization on the overall process. So with the MOC-V retrovirus-like particles, we've put the conditions up here, and this is just to highlight that this is done under a generic protein A protocol. We're not doing anything different to what would be a standard protein A process. We're loading to 80% of the 10% DBC, so high load. The samples are spiked with a 1% volume by volume retrovirus-like particle, and then we're eluting with pH 3.5 and then 3.5 up all the way to 5 with high pH. So you can see by the graph on the right, we're seeing exactly the same behavior that we were seeing in terms of clearance with host cell proteins and DNA. That disassociation between the bound antibody and the impurities that associate with host cell proteins, DNA, virus, is getting greater by being able to elute at a milder pH. You can see with Jet A50 competitor A and competitor B, and Jet A50 high pH, in this case at pH 3.5, the log clearance is standard for what you would expect for the protein A, between 2 to 3 logs. As we go up to pH 4, you can see there's an increase. This still wouldn't be classed as an increase because it's not over 1 log. It needs to be over 1 log to be considered a big increase. But by the time we get to pH 4.5, there's a significant increase, and all the way up to pH 5, where you see almost 5 log reduction value. 5 seems to be the point at which it stops. I think at that above 5, where you have a mixture of not complete dilution and probably virus being associated with the antibody that's not bound, so you're getting low recovery. Actually, you don't see that effect. So I think in those cases, 5 was the point at which we didn't see any more reduction, but there were recovery issues at that point as well with this molecule. So pH 4.5, in terms of everything that we've shown, host cell protein, DNA, recovery, virus, seems to be the optimum point for a lot of molecules. So to support the MOC-V, we're also doing a live virus turn, so we used MLV and MMV. And what I want to highlight here is we've done this in a way that would be done by manufacturers under the regulatory agency's guidance, under exactly the same protocols that a process that the contract manufacturer's customers would go through. And we want to do that for confidence in the analysis, but also to give a realistic expectation for customers when using it, and a realistic expectation in terms of what we're seeing here with the mild pH solution. It's been done under these conditions, and we expect this behavior, obviously molecule dependent. Again, the conditions here highlighting it's a standard protein A conditions. The only two things to highlight on the conditions is with the intermediate wash, we have altered the wash 3 to pH 7 with Jet A50 high pH. Previous talks and information you can find on Jet A50 high pH, the uniqueness of the dilution does give a more rigid wash conditions. You don't have quite the flexibility of DurA protein A, but that's well established. And if you do need any guidance, please just reach out for us. It's in the instructions for use as well for users of high pH. And then we've done pH 3.5 for the standard protein A and gone up to pH 4.5 for high pH to see those differences. And with the live virus, so both MLV and MMV, we see 2 to 3 logs of pH 3.5. We do actually see an increase with MLV with pH 3.5, but it's not significant enough for us to say that there's a clear difference there. However, when we get to pH 4.5, there's a clear log difference between pH 3.5 and pH 4.5 when using Jet A50 high pH. This starts to contribute more to the holistic approach of viral clearance. It supports the relationships that we're seeing with other impurities that would behave very similarly on the resin. So there's a real benefit here in terms of impurities clearance by being able to elute at a milder pH. So I just want to go over the summary of what we've talked about. We talked about DurA Cycle A50, which is our latest generation resin. It is designed as a high capacity, highly stable resin for mAbs, proteins, FC containing molecules, and really trying to address the challenges of complexities, challenges with cleanability. But it also incorporates some of this milder pH solution that you can get with PressureJet at A50 high pH, which I've shown with the case studies can have a huge benefit in terms of impurities clearance for the protein A step. And this can help with process economics. It can help with reduced burden on other steps, and can have a real positive benefit in terms of molecules that previously couldn't use protein A if they were acid sensitive, for example. With that, I would say thank you for listening. I do want to acknowledge all the names that are on the bottom of this slide. Without this team, which is application scientists, field-based scientists, project management, who generated all the data and experiments that I've presented today. And just to highlight something Andy mentioned earlier, with any resin, whether it be DurA Cycle A50, Jet A50 high pH, or other resins within our portfolio, we do have a comprehensive field-based team and technical-based teams to help you. So please feel free to reach out. There's always someone there to help. And with that, thank you very much. Hope there's any questions. If you would like to order a sample of DurA Cycle A50, the QR code is on the screen in front.
All right. And with that, Andy, Mark, this was a great presentation, a lot of great stuff. We appreciate you guys bringing that to us today. And we do have some few questions from the audience coming in. We've got some time for those. Audience members, just a couple reminders. If you do have questions to submit those over on the Q&A panel on the side of your screen. In addition, you should also see our speaker bio panel. It does have information about both of the speakers, including their bios, and it also has their email addresses. So you can just click on that little email address button. You can send them an email or connect with them on LinkedIn as well. And then we also have some additional resources on that page. You should go to download and anything else you might need kind of relevant to the topic. So let's get into the audience questions here. So my first one is about sustainability, and so they mentioned sustainability is becoming a bigger focus for our company. The question is, how is Ecolab bioprocessing supporting its customers to help work towards more sustainable downstream manufacturing?
I can take that one. As you would have seen as part of this presentation and multiple other publications that we've given, we use jetting technology to manufacture agarose beads, and we've gone through the unique properties in terms of mass transfer. But that technology actually results in significant reduction in the use of organic solvents, which is making our process more sustainable, less harmful. And it also then reduces a step. We no longer need to screen the process, which can be a lengthy process. Overall cycle time and carbon footprint is higher. But also it can have the potential to use a significant amount of water. So jetting is an optimized process in terms of water consumption as well. And we're continually looking to improve that process, leveraging technologies that Ecolab already have with water consumption, for example, constantly looking in terms of our processes to make them more sustainable, to support the bioprocessing industry as a sustainable raw material supplier.
Yeah, okay, perfect. Okay. Up next, try to get to a couple more here. So you mentioned a publication regarding the separation of homodimer from heterodimer for biospecifics. Could you talk more about that?
Yeah, I'll be happy to take that one. It was a recent article that was in the Journal of Biological Methods, where they looked at selectivity and binding strengths of one Protein A resin compared to CH1 resins from two suppliers toward a range of sort of standard monoclonal antibodies. And for the CH1 ligands, even though they were targeting the same domain of the antibody, there were some clear differences in the binding strength between the different mAbs, with the widest range being for Presto CH170. The authors then looked to exploit these differences in binding strengths, firstly by just resolving closely related mAbs with pH gradients containing different salt concentrations, and then they applied their observations to successfully optimize a capture step for an in-house asymmetric bispecific, which allowed them to separate these homodimer from heterodimer species. I can check with our marketing if we can send the reference as part of this follow, but hopefully that answers the question.
Yeah, no, I think that was perfect. Let's see. We've got one more we can do here, I think. You mentioned process intensification strategies at the start of the webinar. Which resin from your toolbox would you recommend to use for this?
Yeah, that was me. I can also take that one. Typically, when evaluating Protein A as part of an intensification strategy, there is a desire not only to have that fast mass transfer, as I mentioned during the talk, they want to have the high capacity against a range of different resonance times. But they typically use smaller columns, and these columns are used probably with a higher number of cycles, especially for the manufacture of clinical material. These columns are often coupled to bioreactors or process and feed from high cell densities, so the stability to any potential proteases and the CIP and SIP reagents, that can be beneficial to the longer lifetime of the resin and the cost of goods. So this really was the foundation why we developed and launched DurA Cycle A50. And that would be my first recommendation. However, saying that, DurA A50 and high pH have been used with rapid cycling strategies, so it doesn't exclude any current protein A products, but DurA would certainly be the one I would recommend for a lot of these modern MAP processes. And as I mentioned for CH1, it is possible to have a ligand target in the same general binding site, which offers different selectivity, and it's the same for protein A. All vendors design and manufacture resins in a different way. So the wider screening of different protein A's is definitely becoming more common, especially for more complex formats.
Okay, yeah, great. That's great. Let's see another one here. So what impact does the milder pH elution have on the subsequent purification steps?
I can also take that one. But it's very molecule specific, but as you can see from the talk we've given, there are potentially multiple benefits and there are potentially a lot of reduced burdens on subsequent steps. For example, if you've got a cation exchange step per second and you go in straight into pH 4.5, you eliminate lengthy and sometimes complex pH adjustments, which can also cause stability issues from changing conditions. So there are multiple benefits that this could have on the subsequent steps. And in a very, very unique scenario, it could actually result in eliminating steps afterwards, which then goes on to what we previously talked about in process economics, reducing costs of goods. It's unique, and it just gives a lot more flexibility in terms of unit operations after the protein A step.
Okay, yeah, no, that's great. I think this is actually going to be a good place to wrap up as well. And so again, Andy, Mark, thank you so much for joining us today for your great presentation and getting through these great questions. Audience members, again, we appreciate you as well. Thanks for joining us. And we hope you all have a great rest of your day, and we'll see you next time. Bye, everybody.